Hello everyone, this is Bassi and I welcome you all to the NEET Power Punch series. So today in this video, we will be going to start with the dihybrid cross. We discussed monohybrid cross in the previous session and we will going to recall it very quickly and then we'll complete the dihybrid and some awesome questions. Clear to everyone? So let's get started. Just a second. Okay. Yes. Now my voice is clear to everyone, I believe. Okay. Yes. Now my voice is clear to everyone, I believe. Yes. All clear. Okay, so let's quickly revise the monohybrid cross. What was monohybrid cross? That monohybrid cross was the inheritance of a single character from one parent to the other parent, right? But from one parent to the offspring, or we can say inheritance of a pair of traits from one parent to the offspring. This was monohybrid cross, right? And in the monohybrid cross, we got a ratio, and that ratio was what? That ratio was 3:1. Yes. So in the F1 generation, what ratio did we had? In F1 generation, sir, all the plants were tall. Yes or no? Yes or no? Yes. In F1 generation, all the plants were tall. And in F2 generation, if I talk about the ratio, that is phenotypic ratio, then the phenotypic ratio for the F2 generation was 3:1. And this means what? This means that 3 plants, if suppose in total we have 4 plants, and in those 4 plants, 3 of them is what? 3 of them is tall and 1 was short. Yes or no? This is what we discussed in the previous class. Now, let us move on and let's complete the conclusions made by the monohybrid cross, right? So there is 2 postulates which Mendel gave according to this monohybrid cross. Okay, so what was it? That one is what? The postulates of unit factor. Sir, what is this? The postulates of unit factor. Now, if you're gonna ask me, I am going to ask you one question, that what was unit factor? First of all, I'll ask you what was unit factor. So Mendel considered that genes are the unit factors. Okay, so he gave a postulate which was unit factor, and the second postulate he gave was postulates of dominance, and the third he gave was postulates of segregation. So in total, on the basis of this particular monohybrid cross, he gave 3 postulates. Okay, sir. Now, here, what is going to happen? These 2 postulates made the first Mendel's law, and this is known as law of dominance. So both these 2 postulates made what? Made law of dominance. Now what? Now, sir, postulate of segregation, it made law of segregation. It generated law of segregation. Clear to everyone? So according to the Mendel's experiment, we have 2 laws, law of dominance and law of segregation. Now let us understand these laws of dominance. Now see, the law of dominance is what? It is used to explain the expression of only one of the parental character. Yes, you heard it right. When we go back to the cross, so we saw that we took pure hybrid parents during F1 cross. Yes, sir. One parent was totally tall and the other parent was short. Okay, clear to everyone? That's great. So here, what happened when they formed the offsprings? They got a new character, new type of genotype, I should say, which was capital T and small t. Clear? But in this, what was the phenotype in F1 generation? What was the phenotype in F1 generation? So your answer will be that the plant was still tall. The plant was still tall. all the plants produced in what? In the F1 generation was tall, right? That means what? That means something, the short plant is not able to show its character. This means this only. If I have short plant and I have tall plant, and if I make them reproduce and a tall plant is only getting produced, so can I say that the short plant is not able to express its characters? Yes, sir. So the character which is expressed here is tall. So can I say that tall was the dominant character here? Yes or no? Yes. So the law of dominance said that among two traits, one will be dominant, and that will be the one whose phenotype is going to be expressed, right? Let us understand with the help of the F2 generation also. So we saw that during the selfing in F2 generation, that is capital T and small t, what we got? During this, what we got? We had the gametes, which is capital T, and we had this gamete, which is small t, right? Capital T and small t, we had the gametes. Here also we had capital T and small t. And in F2 generation, we got a ratio of 3:1, sir. Yes, obviously. And this F2 generation proves the law of dominance more. It gives a more clear idea of the law of dominance. How, sir? See, 3 plants were tall, okay, and 1 plant was short. But when we saw the genotype of this thing, so in this, 2 plants were like what? 2 plants were having capital T and small t. 1 plant was having capital T, and 1 plant was having small t, right here. So this was the ratio which we have got, which was 1:2:1. So among here, this two plant doesn't have only dominant gene, it had recessive gene also, but then also the character shown was the tall. So that means tall was the dominant here. So this is how Mendel proved that among 2 alleles there is one allele which is dominant and which is represented by the capital letters, and it is able to show its phenotype in heterozygous condition also, in presence of any other person also, it is able to show its character, and that's what is dominant. But talk about this small t was present here, but it was not able to show its characters, and that's why we call it as our recessive allele. Is it clear? This is how he gave the law of dominance. Clear? Now let us move on and let us talk about some of the exceptions to this law of dominance. Yes, biology is our science of exception, and we have lots and lots of exception here. So the exception for this case is what? It is incomplete dominance and co-dominance. So there are 2 conditions which we are definitely going to learn, but not today. After, in coming few classes, we are able to learn. So exception was what? One is incomplete dominance, and the other one is what? The other one is co-dominance. So if someone asks, or if a question comes that what is the exception, or which thing among which of the following does not follows the Mendel's law of dominance, so you will write incomplete dominance and co-dominance. What are they? We will see it. We will definitely see in some coming classes. Now, next is what? This was the law of dominance. Let's now go to the law of segregation. Now, sir, what is this law of segregation? Segregate means what? To set apart, right? To break apart. Yes. So here Mendel said what? Mendel said that if they are talking about the gametes, if we are talking about the gametes, okay. So what happens? Let us take an example. This is a cell, and inside this cell we have a chromosome, okay. And remember, whatever I am drawing right now is 2n. And if you have any confusion, what is 2n, what is n, how does division takes place? So right now in droppers batch, I have completed mitosis, one shot mitosis I have completed. And if you have any doubts in those things, go to the lecture and watch that video after this class. The concepts of mitosis will be very much clear to you. Okay, now, so here we have what? Here we have this chromosome. Okay, right, just a second, I'll change this thing. I have one chromosome and let's suppose that this is its pair. So what happened? White chromosome came from mother, or pink chromosome, let's suppose, came from the father. So they are the homologous chromosome. Yes, sir. This is what? This is a normal cell. This is what? This is a normal cell. Normal cell means our normal cells, maybe hand cells or anything. And if I talk about with respect to plant, so this is a plant's normal cell, maybe leaf, maybe stem. But when we talk about the gamete formation, so the germinal epithelium in our body makes gametes, right? So during gamete formation, what happens? This particular body is 2n, yes or no? Yes, obviously, sir, because it is having pairs. The chromosomes are in pairs, right? So if it is 2n and then this particular thing is going to make the gametes, so gametes will be haploid. Yes, very good. Gametes will be haploid. So how it will happen? What is going to happen? So during the gamete formation, white chromosome will go in one gamete and the pink chromosome will go in the other gamete. That means what? That means there is the segregation of the homologous chromosome. There is what? Segregation of homologous chromosomes. So during the gamete formation, homologous chromosomes get segregated. This is what? This is one of the laws given by the Mendel. Okay. And this law is also known as law of purity. Now let's understand what things were written here in the laws of segregation. So let me find the, yes. So the statement says that unit factor of one pair, unit factor, which means genes of one pair, segregate from each other. See, if we are drawing here, these were the homologous chromosomes, right? So this was the unit factor. So here white will go in one gamete and pink will go in another gamete during the gamete formation, and only one unit factor, only one gene, only one allele related to each other characters move into a gamete. Let's suppose that this thing controls the plant height. So let's see this suppose this is the capital T and this one is the small t, right? Okay, so both the chromosomes, since they are homologous chromosomes, so both the chromosomes will going to control a single character, right? So the statement was saying that only one of its type or only one of its allele will go into the gamete. So here we have capital T and here we have small t. This is the law of segregation, that homologous chromosomes segregate during gamete formation. I think so it's pretty much clear to you all now. Now let us move on and let's again get back to that statement. Where is that? Oh my god, did I lose it? No, it's here. See, now it is also referred to as law of purity, which I right now said, of gametes, as gametes are always pure for a character. How, sir? How? What you said right now? See, in a gamete only one allele is there. Yes, in a gamete only one allele is there, right? So there is only and only one allele, whether it will be capital T, whether it will be small t. So I can say that gametes are always pure, right? Either capital T or small t. So gametes are always pure with respect to a particular character. Clear? This was the laws which was given by the Mendel. But again, before moving forward, we have an exception. We have an exception, and that exception says non-disjunction of homologous chromosomes. There are some cases during which the gamete is formed, but these homologous chromosomes couldn't segregate from each other, and this creates genetic problems, genetic diseases, and lots of things. But right now we will be focusing if non-disjunction of homologous chromosome is happening, then it is an exception to the law of segregation. How, sir? How? What is this, sir? Please explain. See, this is your cell, okay? Or else I will not draw cell, I'll just draw the chromosome, okay? Suppose this was the white chromosome. Where is the white color? Yes, here we have. This was the white chromosome and this was the pink chromosome. They both were homologous, okay? Now they want to make the gametes. Yes, sir, they want to make the gametes. So now how they are going to make the gametes? In this, what happened? They made two gametes. Obviously, two chromosomes will be there, then two gametes will be formed. But they were not able to segregate from each other. And if they are not able to segregate each other, only one gamete, the one gamete is going to have both the chromosomes. One gamete is going to have both the chromosome, and this gamete will have nothing. So this is called as non-disjunction of chromosomes. Okay, clear? Exception to the law of segregation. Exception to the law of dominance was what? Exception to the law of dominance was the codominance and incomplete dominance. Right? Now, we have cleared everything about the monohybrid cross, and if you have any kinds of doubt related to monohybrid cross, what you have to do? Comment down below. Now, let us move on with the gamete formation. This is a very crucial step if you want to go with the dihybrid cross. So before that, I would like you to understand how we make the gametes. Clear? Okay. So gamete formation is basically what? Let's suppose I am going to give you a genotype. Okay. I am going to give you a genotype. The genotype is, let's suppose, capital A and capital A. Okay, sir. Now, this first step will be what? First step will be identify as homozygous or heterozygous. Okay? First step will be to identify as homozygous or heterozygous. See, we have A and A, right? We have A and A. Both A are capital. Yes, both A are capital. And if both A are capital, then that means what? It is homozygous. Yes, sir, it is homozygous. Great. It is homozygous. Now what, sir? Then if it is homozygous, then try to divide it. Okay, try to divide it. Let's suppose this is your A and A. So it will go in one gamete and it will go in another gamete. So if I ask you how many types of gamete formed, I'm not asking how many numbers, I'm asking how many types of gametes. So you're going to say that, sir, we have two gametes, but both are of same kind, right? So whenever we have the homozygous condition, types of gametes will be what? Types of gamete will be 1. So let's make a table and understand every condition, okay? We are going to make a table and let's make this gamete formation very clear. It will be very easy for you to do the dihybrid cross, trihybrid cross, whatever the questions, ratios, questions and all, it will be very easy if you know this. So that's why I am teaching you. Now see here, we have the genotype, okay? In the one hand, we'll write here genotype. Why I am writing the spelling of genotype wrong? Genotype, okay, leave it. Next we have number of heterozygous pair. What we have to search? Number of heterozygous pair. Okay, okay, sir. Next, what we have to search? Next, we have to talk about the types of gametes. Next, we have to talk about the types of gametes. Clear? Okay. Now what? Now we are going to talk about the different kinds of genotype. So here we have capital A and capital A. We have capital A and capital A, which we saw. So number of heterozygous pairs are what? 0, because this one is This one is homozygous condition. How many types of gametes? Sir, how many types of gametes we are going to get? We are going to get one type of gamete. Okay, we are going to get a single type of gamete. Okay, great. This was very easy. Next, what? Next, hi OP. I'm glad that you are here. Let's study. Next, what? Next is the capital A and small a. Capital A and small a. Now this capital A and small a, if I ask you to make the gametes, if I ask you that how many types of gametes are here, so what can you do? You are going to find the number of heterozygous pair first. So how we are going to find? See, this is capital A and this is small a. So this pair is the heterozygous pair. Both alleles are different. So I'm going to write I have one heterozygous pair. Okay, sir. Now, just imagine if these heterozygous pair is gonna make the gametes. So one gamete will have capital A and the other gamete will have small a. So how many types of gametes we have got? We have got two types of gametes. Clear? Now let's make this thing little more hard. Let's have capital A, small a, capital B, small b. Okay, sir. Done. We have the another one which is capital A, small a, capital B, small b. So in this capital A, small a, what is going to happen? See here. How many pairs of heterozygous pairs are present? Is it one? Is it two? How much? See, this is one heterozygous pair and this is one heterozygous pair. So in total we have two heterozygous pair. So how many types of gametes we are going to get? We are going to get four types of gametes. How, sir? How we are going to get four types of gametes? Let's see here. So capital A, small a, capital B, small b is present. Okay, capital A, small a and capital B, small b is present. Now let's try to make gametes. So this capital A and small a will make what? Will make a kind of gamete, and this capital B and small b will go into another gamete. So let's suppose here we have what? Here we have capital A, capital B, because both are what? Both are we have this two alleles here, like two genes here. So this A and this B can come. Okay, and what? This A and this B can come. Okay, sir. This A and this B can come. Okay, and can I say that this A and this B can come? Okay. So how many types of gametes which we got? We got four types of gametes. How many types of gamete we are able to make here? Sir, we are able to make here four types of gametes. So let's write in the sheets. Okay, clear? Now let's move on and let's make this thing little more difficult. Okay, little more difficult, or maybe little tricky, right? See, next is what? Next we have is capital A, okay, small a, capital B, small b, capital C, small c. Clear? Okay, sir. How many heterozygous pair do we have? We have, sir, three heterozygous pair. And if you are having three heterozygous pair, then how many gametes you are going to make? See, you are going to have 8 types of gametes. How many types of gametes? 8 types of gametes. Clear to everyone? In the similar manner, like if you have so 2 gametes. If you have 2, 4 types of gametes. If you have 3, you have 8 types of gametes. Clear? Yes, sir. But this is kind of confusing, sir. I'm not getting it properly. So to avoid the confusion, we have another method, and which is very easy, which is known as fork line method. In this, you don't have to worry about anything. This is known as what? This is known as fork line method. And how you're going to found gametes in this thing? How you're going to form gametes in this thing? Let's check here. Is everything fine? Am I audible? Am I visible to everyone? Just let me check once. Yes. Okay, I think so. Is there any lag? One second. Okay, just wait for a while. Everything will be fine. And I think so. We are back again, right? Yes, we are back. Definitely, we are back. Okay, so let's go with the fork line method. What is fork line method, sir? It's a very easy way of finding the gametes. Very easy. So in this fork line method, I'll give you an example. Let's start from basic. So we have capital A and small a. Okay, we have capital A and small a, clear. So what you have to do is that you have to make two lines. If you have heterozygous type, you have to make two lines. Diversify it, bifurcate it. So one you write capital A and the other you write small a. How many types of gametes you got? One gamete, two gametes. So what is the number of gametes here? Two types of gametes. Okay, clear, sir? All okay. Two types of gametes. Now, next is what, sir? Next we have, let's take another example. Let's take another example. Am I live? Is there any issue with my stream? Just let me know in the comment section. Are you able to hear me? Are you able to see? Just a second. Okay, now let me know that am I visible? Am I audible to everyone again? Let's see. Let me check. Let me check that. Yes. So we are back. We are back. Okay. So what we were discussing about the gamete formation through the fork line method, right? So in the fork line method, what we have to do is that if we have two things, like we have this allele, we have this genotype. So in this genotype, first of all, you need to figure it out whether this is homozygous or heterozygous. So it is heterozygous, right? So if it is heterozygous, bifurcate them into two. So we have A and capital A, and this is will be your gametes. Just you need to bifurcate and your gametes will be ready. So we have two types of gametes. Clear, sir? Now, what to do in this case? What to do in capital A and capital A small a and capital B small b case? So here what we are going to do is that here again we are going to do something like this. See, I'm gonna take the next page and let's write here capital A, capital small a, and capital B, small b. So here you're going to bifurcate like this. First of all, bifurcate them. Okay. So, okay, sir. We will bifurcate them. We have capital A and we will be having small a. Done. Now what you have to do? See, this is the crucial step. Now this is also what heterozygous condition. So bifurcate here capital B, small b, capital B, small b, and now make the gametes. How, sir? See here, very easy. This will be your one gamete, which will be AB. This will be your another gamete, which will be capital A, small b. This will be your another gamete, which will be small a, capital B, and this will be your another gamete, which will be AB. So this is how you're going to get the genotypes of each and every gametes which you are making. Okay, sir. Mazara, give me the other question also. Okay, definitely I'll give you. What question do you need more? Let us try to make the another one. Which one, sir? This capital A, small a. Okay, small b, and then capital C, small c. How you are going to make this? It's very easy. I'll show how you can make. Okay. So here let's have again, let's diversify them. So first of all, let's make this capital A, small a. Clear, sir? Now, but this thing is what? This thing is not heterozygous. This is homozygous. And if this is a homozygous condition, what we are going to do is that we are just going to write B here and B here. When we are going to do this, when there is an homozygous condition, in homozygous condition, we are not going to bifurcate it. We are just going to write it B and B with the previous heterozygous condition. With the A, we have written B and B, because in both the cases, small b and small b will only be present, right? Next, what we are going to do, we have C. Yes, sir, we have C. Is it heterozygous? Yes, sir, it is heterozygous. So now what? Now we are going to bifurcate it again. Okay, so this is capital C, small c, capital C, small c. I'll move for a little bit side so that you can see. Capital C, small c. Now, how we are going to get the gametes? We are going to get the gametes here like A, B and capital C. We are going to write, get the gamete as A, B and small c. See, this A, B and C will be there. Okay, this A, B and C will be there. Clear? And now next, what we are going to have, we are going to have A, B and this C, and then we are going to have A, B and this C, right? So how many types of total gametes did we have? We had 4 types of gametes with different gamete genotypes, right? This one is capital C and this one is the small c. I think so that this is clear to everyone. Clear? This is how we are going to make the gametes, right? Okay, sir. Gamete formation is done. So in the fork line method, you have to remember that for fork is not created for homozygous condition. Fork is not created for what? For homozygous condition. Clear? Homozygous condition, fork is not created. There is no need of creating the fork. Clear? Just keep this thing in mind. Rest, everything will be fine to you. Now, here comes the dihybrid cross. Now let us move on towards the dihybrid cross. We have done the basics which is required to understand the dihybrid cross. So let us go with the dihybrid cross, sir. Okay, how we are going to do this dihybrid cross? Let's see here. So here, if the dihybrid cross, first of all, let us understand what do you mean by dihybrid cross. So dihybrid cross is the cross in which we are studying the characters, 2 characters, like how 2 characters is moving into the offspring. Let's suppose I want to see that whether my eye colors or my hair color is related or whether they are not related, or how these two, both of them goes into the other like offspring, right? So if this thing you have to study, we will be performing dihybrid cross. So dihybrid cross is basically what? Dihybrid cross is basically the cross performed to study the inheritance pattern of 2 characters. Okay, cross performed to study the inheritance pattern of 2 characters together. Or can I say that cross performed to study the inheritance of 2 pairs of contrasting characters. Clear? Okay, sir. 2 pairs of contrasting character means what? Let's suppose we are talking about the tall plant height. So one will be tall plant, the other will be short plant. Clear? Okay. So we are talking about 2 pairs of contrasting characters. Now, let's move on. In this, we will be taking 2 characters. Now, sir, why we are taking 2 characters? Because Mendel has taken, that's why. So seed color, do you remember that trick, fat Ria 1? So seed color will be what? Seed color, dominant seed color will be yellow. Yes, sir. So let's write here. So seed color we have capital Y, which will represent the yellow. And should I represent the green with capital G or G? No. If we are representing the recessive, we are going to represent with the same alphabet, but in smaller case. So small y represents green thing. Okay, sir? Clear? Now what? Now we are going to talk about the seed shape. So seed shape, how this seed shape is going to have the dominant character. So the dominant character for the seed shape is round, and we are going to represent it as capital R. Okay, and if I'm talking about the recessive character of the seed shape, so it is what? It is wrinkled. So should I write with W? No, sir. We should write with the small r. I think so. These things are pretty much clear to all of you. Okay. So characters are clear, seed color is clear, seed shape is clear. Now what we are going to do is that we are going to study like how this seed color and seed shape are related, whether they are related also or not, or whether they are completely related, or whether like dominant seed color and dominant seed shape goes together, or whether dominant and recessive seed color can go together. All the things we are going to study. So for this we are going to perform a cross. Okay, again, what type of cross? In all the crosses we have seen that what we have seen that Mendel took what? Mendel took the pure line. Okay, so we are also going to take pure parents. So pure parents will be what? It will be capital Y. I will be writing capital Y in italics and small y in cursive. Okay, so capital Y and capital R. So this creates what? This is basically the dominant parent, right, sir? Yes. How dominant? We have capital Y and capital R, which is what? Which is basically representing yellow color and round shape. Okay, so let's write here yellow color. Okay, so we were discussing that this will be what? This will be yellow color. This will be yellow color and round shape. Okay, in the similar manner we will be having capital Y and small y and small r. This is gonna represent what? The recessive character, which is what? Which we have is green color and wrinkled seeds. Okay, green color and wrinkled seeds. I hope that I am clearly audible and visible to each and every one of you. Okay, yellow color. Now what we will do is that we will end this session because I am facing some technical issues, and in the next session we will be going to complete